I'm going to speak a little bit about a novel platform for a single cell detection of multiple CRISPR-edited gene modifications. I'm an instructor in Kathy Wu's lab at Dana Farber. One of the long-standing interests of our lab is the study of chronic lymphocytic leukemia, which is the most common adult B-cell leukemia and is characterized by a high degree of genetic heterogeneity. And despite notable advances in the clinical management of CLL patients, thanks to the introduction of target therapeutics, CLL still remains incurable. And clonal evolution and accumulation of high-risk genetic mutations are frequently associated with resistance to standard and novel therapies. And in fact, despite the wealth of knowledge that we have gained through large-scale human next-generation sequencing studies, we still incompletely understand the relationship between genotype and phenotype of CLL cells carrying certain genetic traits, and how this in turn influences B-cell function and impacts disease progression, transformation, and response to therapy. And this is because of the many challenges in CLL functional genomic studies that we've been facing throughout the years. First of all, the fact that primary B cells are very difficult to manipulate in vitro. The primary CLL samples only show short-term engraftment in vivo xenografts, and that there's also a limited number of primary CLL-derived cell lines and mouse models that can faithfully recapitulate human genetics. But thanks to the introduction of novel genetic engineering technologies, including conditional knock-in and knockouts, and more recently CRISPR-Cas9 gene editing, now we're really provided with an unprecedented opportunity of modeling a single or multiple alterations in a cell context of interest better than previously possible. And that's why we undertook the use of these novel technologies to develop novel mouse models of human CLL genetics, starting from the generation of a mouse model carrying combined point mutation in the splicing factor SF3B1 and deletion in the DNA damage response gene ATM, which only when combined, and this is a frequently co-occurring alteration also in human samples, these animals were able to develop disease at a very late onset, which is consistent with the indolent nature of human leukemia. And more recently, I was also involved in an effort that we're about to conclude, focused on the generation and characterization of another novel mouse model based on point mutation in the B-cell developmental factor IKZF3, which as a sole lesion and in heterozygosity was able to induce leukemia in animals, again, with a late onset. And further, what I focused a lot of my efforts over the last 2 years is to generate in vitro and in vivo tools for the functional interrogation of common loss of function lesions, including in genes involved in DNA damage, such as TP53, ATM, and SAMHD1, BERK3, as you can see in the red boxes, which is involved in NF-kappa B signaling, MGA, which is a negative regulator of MYC, and CHD2, which is a chromatin remodeler. These are the most common loss of function mutations of human CLL, which we are able to model efficiently through CRISPR-Cas9 gene editing. By means of how Cas9 works, in the presence of a sgRNA targeting a sequence of interest, Cas9 can introduce double-strand breaks, and upon the non-homologous end joining a repair of the target lesion, there is insertions and deletions at the DNA target sequence, which most frequently induce loss of function of the gene of interest. And this is what we utilized to model these lesions, either individually or in multiplex fashion, both in cell lines and more recently in mouse models. But before I dive into the description of our data and the novel tools that we've developed, I would like to acknowledge that this was a very close collaboration with the group of Luca Pinello at Mass General, and as you've mentioned, Kendall Clement, who's also connected with us today, who's developed all the computational pipelines for analysis of both bulk and single cell sequencing data. And then I've also closely worked with Shichang Li and Ken Levac from the Translational Immunogenomics Lab at Dana Farber, and with Dave Ruff, who has introduced us to the use of Tapestri. So to move further, what I first will describe to you is the generation of in vitro models. We have used this widely used Probe cell line, BAF3, which we have first engineered to constitutively express Cas9 and then further transduced with lentivirus expressing sgRNAs against the individual genes of interest, in addition to an mCherry marker to be able to track these cells by flow cytometry. And what we first did was to verify presence of gene edits through a bulk targeted PCR-based next generation sequencing approach, followed by identification and quantification of insertions and deletions through CRISPR-edited software analysis. And this is the software that Kendall developed. And as you can see in the example below about TP53, there's a number of deletions and point mutations that occur across the predicted cleavage position for the selected sgRNAs. And through these analyses, we're able to confirm efficient editing of the 6 target genes across the 6 individual cell lines. But we wanted to use these tools for single cell interrogation of our ability to introduce gene edits, and not only on the 6 on targets, but also to explore off-targeting activity of Cas9. And that's where we partnered with Dave Ruff and Xu Wang to design a targeted single cell detection panel for the 6 on targets and the 22 predicted off targets, or in silico predicted off targets of these sgRNA through the CRISPR algorithm. And the panel also included 12 internal positive controls for analysis. And I'm sure the audience is familiar on how the Tapestri work, but essentially each cell is uniquely barcoded. And so that upon analysis, we're able to deconvolute each cell and each amplicon. And within each amplicon, Kendall was able to run the CRISPR software and compute the percent modified reads. So just a few QC steps here. I'm showing you that we've obtained a sufficient coverage and efficient coverage across all the targeted regions. And these numbers were even higher when with some of our latest runs performed on the newer version of the Tapestri machine. So our first test run was essentially to combine equal proportions, more or less equal proportions of the 6 single edited lines and run them through the Tapestri. And as you can see, we're able to analyze roughly 3,400 cells, and we're able to identify modifications and very high efficient modifications. As you can see here, the percent modified reads was almost 100% in most of the cells in discrete proportions of cells. Again, these are single edited lines that are mixed together, whereas off-targeting was negligible. And what Kendall also did, which I find to be a very clever approach, is to understand the zygosity of our calculated alleles, essentially computing the abundance of the 4 possible assortments of wild type versus edited alleles. As you can see, we can compute presence of cells carrying both wild type alleles, or cells carrying one wild type and 1 mutant alleles, and also cells carrying either homozygous mutant or heterozygous mutant. And this is particularly relevant in the context of studies of cancer evolution that we are currently performing. But of course, I'm a cell biologist. I'm interested in function. So I was interested in understanding whether these single lesions alone could confer survival advantage to the cell lines. And what is commonly used to assess tumorigenicity of cancer drivers in the BAF3 line is the IL-3 withdrawal assay. So essentially, removing IL-3, which is a growth factor for BAF3, we're able to test whether any of these loss of function mutations are able to still facilitate survival and proliferation. This was the case. Indeed, all of the 6 cell lines proliferated and survived in vitro better than the Cas9 control alone. But we all know that cancer is definitely not arising from a single mutation alone, but it's rather due to a complex interplay of co-occurring lesions. And that's why we undertook the generation of multiplexed approaches. And in this case, we multiplexed our lentiviral transduction strategy and concomitantly introduced all the 6 gene edits by pooling lentivirus expressing all 6 sgRNAs. And as you can see, again, confirming first by PCR-based targeted NGS, we were able to see edits across all 6 genes in lower proportion than the single edited lines, but still present across all the 6 target lesions. And what Ken Levac devised here is an approach by which we were able to quantify presence of individual or multiple sgRNAs in single cells through a qPCR-based method ran through the Fluidigm biomarker platform. What we did, we sorted out mCherry positive BAF3 lines 3 days post-transduction. Again, mCherry is a marker for sgRNA presence in 96 well plates, which were then tested by single-cell qPCR. And as you can see, we observed roughly mostly 2 to 3 sgRNA in each individual cell, which were even higher in some of the latest studies that we've done with higher titer lentivirus. But again, presence of sgRNAs only tells us, only partially tells us the potential of gene edits to occur at the single cell level. And that's why we ran these multiplex cell lines through the Tapestri. And we're able to analyze 3,200 cells across all the 6 on targets. And as you can see here from this subset plot, we were able to verify presence of multiple distinct co-occurrences across all of the 6 on targets, and in different proportions. And again, we're interested in function, and we're interested mostly in interrogating which of these genes can co-occur together and which of the co-occurrences facilitate in vivo survival. And that's why what I did, I transplanted this multiplex line into immunodeficient NSG recipients and sampled it again when animals reached survival. Essentially, I harvested splenocytes from NSG animals, sorted them for cherry positivity, and ran them through the Tapestri. And as you can see, we observed a very strong selection of cells carrying combined mutation in MGA and CH2, which is what was an unexpected combination of drivers, but very interesting to see in this setting. Finally, a quick, so I developed these tools with the intention of modeling these lesions into mice because I'm interested in understanding not only the function in normal B cell development, but also in leukemogenesis when these lesions are introduced in a multiplex fashion. And that's why we were lucky to be able to access a mouse model that was generated in 2014 that carries conditional expression of Cas9 together with the GFP marker, which we were able to cross with CD19 expressing animals to generate a CD19 Cas9 line. Essentially, in these animals, Cas9 is expressed exclusively in B cells under the control of the CD19 promoter, which is expressed, CD19 is turned on early during B cell development in the bone marrow. And what I did was to devise a strategy by which hematopoietic stem and progenitor cells from CD19 Cas9 donor mice from young animals are isolated from the marrow, transduced in vitro with lentivirus expressing single sgRNAs against the genes of interest or a non-targeting control, and then retransplanted them into irradiated immunocompetent recipients. And by doing so, I was able to generate 7 strains of animals, 6 of which carrying individual gene edits, and 1 which I will label as NT1, which serves as a non-targeting control. And the unique feature of these models is that we're able to track edited B cells in the peripheral blood of these animals by flow cytometry because of expression of Cas9 GFP and of expression of the sgRNAs, which instead carry the mCherry marker. So what we can do is isolate B cells, which carry sgRNA, so edited B cells, and through the same strategy as we did before, we were able to verify presence of gene edits restricted to the B cells of these mice, but not in other lineages. And also, we also sorted GFP cherry positive splenocytes from 6 separate single edited mice and ran them through the Tapestri. And we're able to also verify that even in our in vivo system, editing was restricted to the on targets, whereas off targeting was again negligible, which was quite reassuring to see, considering that we all aware of how detrimental can long-term effects of Cas9 be in an in vivo system. And the unique feature of these models, as I partially mentioned, is the fact that we can isolate edited and unedited B cells from the same mouse, which allows for paired comparisons. And this is why we sorted edited and unedited B cell splenocytes and profiled them by RNA-seq from 3 animals per group. And as you can see, we were able to observe a number of commonly dysregulated genes, either upregulated or downmodulated. And when we interrogated the pathways that were commonly modulated across these 6 strains of animals, we observed, and possibly not too surprisingly because we're dealing with oncogenes, but interestingly, we did observe pathways that were involved in cell survival, DNA replication, and signal transduction and metabolism to be commonly dysregulated. And functionally, what I did observe was that upon in vitro culture with the mitogenic stimuli, LPS and IL-4, B cells from any of the 6 strains carrying individual gene edits were able to proliferate and survive better than the non-targeting control. Again, confirming what we had previously seen in cell lines, that each individual trait is able to confer some survival advantage, even at the normal B cell stage. And now I'm exploring multiplex mouse models, which I'm not able to share right now, but I will be sharing at the upcoming ASH meeting, which are providing us with incredibly exciting new avenues for interrogation of the function of these drivers in leukemogenesis and even in transformation into a more aggressive lymphoma. So to conclude, I hope I convinced you that we can generate in vivo and in vitro modeling of common loss of function mutations of CLL through CRISPR-Cas9 gene editing. And then through the Tapestri system, we were able to not only verify presence of our edits within the on-target regions that we have modeled, but also we were able to interrogate co-occurrence of gene edits in multiplex systems, and also to functionally interrogate co-mutation upon in vivo transfer with the new and co-occurrence of MGA and CHE2 as being 2 lesions favoring survival in vivo. And finally, we're applying these tools to mouse models, and I'm hoping to be able to share some of that data soon. So with this, I would like to conclude by thanking my mentor, Kathy Wu, who has given me a lot of freedom in the exploration of novel tools for functional genomic studies in vivo models, and all the other names on this slide who have been instrumental for this and current projects. And I also would like to acknowledge support from ASH and from the Leukemia Lymphoma Society. And thanks for the attention. I'm happy to take questions.
Thank you so much, Lisa. This was wonderful. So I'd love to take the first question. How did you decide where in the gene to make the edit, as opposed to making multiple edits across the gene?
Yes, so we have tested, we actually in our initial studies that were performed by Maria in the lab, was also on this acknowledgement slide. She's now back in Spain, but she did a lot of work also in human cell lines and also in these first mouse studies. We have tested 4 sgRNAs per gene, and we focused mainly on the very early exons because we wanted our mutation to be truncating and to be disrupting the functionality of the gene itself.
I see. So are they correlated with what you would find in clinical human samples, or are the clinical human sample mutations also loss of function and you just went with a loss of function strategy?
Yes, so all these lesions that we have modeled through CRISPR-Cas9, we definitely focused on the loss of function. What ATM and TP53 are, they are commonly deleted. They can also be mutated, but also commonly deleted. MGA is also a truncating mutation. Actually, MGA was also found to be frequently, there is an intronic polyadenylation, which facilitates truncation of the protein. And similarly, all of these are loss of function in human samples.
Great. Okay, so we have a couple questions. Let's see here. So someone was surprised to see very few off targets, and they noted that you were also surprised as well. When you're designing a Tapestri panel and looking for off-target events, how many would you suggest interrogating?
So we also, in that case, we focused on off targets that would potentially have an impact on B cell function. So we focused on exonic off targets and also on intronic off targets, but we chose intronic off targets in genes that are involved in B cell development, such as I do recall one of the SAMHD1 off targets was within MUN1, which is IRF4. It's a very important B cell developmental factor. We did choose 2 to 3, if I do recall correctly. Yes, we chose 2 to 3 off targets per on target. Plus additional intronic. For sure, I know that Kendall, and maybe you can comment more on this, is doing a much broader study right now on human cell lines, interrogating multiple off targets to, I think I can call on Kendall to comment on this. Kendall?
You're on mute.
Sorry about that. Yeah. Ultimately, I think this is a very important research question that's probably open-ended. I mean, we're still trying to figure out exactly how many you have to profile, but it definitely, it's an important question that we're definitely looking into. So important.
So we have a question for you. Someone wants to know, were the bioinformatics difficult for single cell? Are there tools to analyze these data at the single cell level, or did you create your own? And if you did create your own, are they publicly available?
Yeah, so definitely this is we use CRISPR, as Elisa mentioned, to quantify the editing, but kind of compiling the results and getting it all to work through CRISPR is kind of custom stuff. We're planning to again publish in the near future, hopefully.
Great, thank you. Question, what is the upper limit of how many genes you think you could look at? So you did 6. Are you planning on doing a bigger panel?
And not at the moment, because we are extremely busy in trying to characterize multiplex models that we obtained through these 6 genes. But there are ongoing efforts on the human side where they're extending the number of samples that are analyzed through combined cohorts from here and from Europe. And there's definitely novel disease drivers that are emerging. Of course, with this strategy, we would again be limited to the interrogation of loss of function. But of course, with novel CRISPR-Cas9G strategies, we can also interrogate better point mutations. Right now, there's a lot of excitement for our leukemia models. So not in the immediate future, but definitely we are thinking about exploring these platforms further.
Great. Technical question. After flow sorting, what percentage of the cells had the marker for lentivirus?
Yes, so we used very high titer lentivirus. So I would say in the single edited cell lines, we are able to see up to 60 to 70% positivity upon a transduction and in vitro culture for around 3 days. When I did this in my mouse models in hematopoietic stem cells, those are more tricky to transduce, but again, with very, very high titer lentivirus, we were able to obtain roughly 50% cherry positivity upon 3-day in vitro culture.
That's phenomenal. Things have certainly progressed over the last several years. And then I think we have one more question unless other people want to jump in. The question is, does the zygosity matter for these genes, whether it's a heterozygous or a homozygous loss of function?
I am seeing in our current experience with mouse models, we're seeing that invariably homozygous mutants or heterozygous double mutants tend to accumulate over time. I do think that having both copies mutated may facilitate the production of the aberrant protein better, because we invariably see selection for cells carrying 100% modified alleles. So there's definitely a functional implication for that.
Wow, very interesting. All right, well, if there are no further questions, Elisa and Kendall, thank you so much for joining us.
Thanks for having us.
Thank you.
We look forward to your publication.
Thank you.
All right, bye.
Bye, have a good day.